Good afternoon, class, and I hope you all are having a great week. And this is a good day because this is our final lecture for this semester. And in this lecture, we are going to be discussing the world of microbes. That's what we're diving into. And more specifically, we are going to take a look at Staphylococcus aureus. Let us begin. First, we're going to cover the basics of Staphylococcus aureus, and it is known as golden staph, and it is known as those individuals who, it is known by that by individuals outside of the scientific community. And this microbe is one of the most common types of bacterium, and it mostly resides on the skin or inside of the human nose. Good, right here is a little microscopic picture of S. aureus, and as you can tell, it has it's bundled up in a little sections and clusters, and we're going to discuss those clusters and what they're called later on in this lecture. Now we're going to discuss the taxonomy of S. aureus. But first, we need to discuss what taxonomy is. And taxonomy is, oh, I'm sorry. I apologize, is a type of science that involves classification, identification, and nomenclature of living organisms. And it is organized into seven different levels. And these levels are domain, kingdom, phylum, class, order, and let's see, the order, family, genus, species, and subspecies. Um, S. aureus can also be difficult to classify, and this is due to the sheer amount of different species of Staphylococcus that exist in the world and how similar they are to each other. Um, Most microbiologists will look closely at these characteristics to classify them in each different level. And here we're going to discuss what of the each of the classifications for S. aureus are. So S. aureus belongs to the kingdom Bacteria, the phylum Bacteria. I'm sorry if I mispronounce this. Bactili Bactylota, class Bacilli, order Caryophanolus. The family is Staphylococci. The genus is Staphylococcus. The species is the same name as the genus Staphylococcus, and the subspecies is aureus. Now, the very specific thing about the subspecies is that the subspecies, they all share the same traits, and these organisms interbreed with each other as well. To the right here, you can see a simplified version of the taxonomic classifications with the family, genus, and the species. This also lists two different species of Staphylococcus as well. Next, we are going to cover the range of S. aureus. And S. aureus has a wide host range, and it spreads across several different species of organisms as well. The largest reservoir and the main niche of S. aureus are the nares that reside inside the human nose. And according to different studies that have been found, there is an average that one out of every three people is colonized with this microbe, making the infective number inside the entire world to be about 2 billion people at one time. The second largest carrier of this microbe is farm animals. More specifically, cows are the largest carrier in the farm animal world. And through several studies that has been conducted, it's been found that this microbe has a high prevalence in the mammary glands of dairy cows. So, and by the use of these studies and by using the population of cows in the entire world, it has been calculated that an estimated amount of about 75 million different cows can be infected with this microbe at one time. And to the right here is just a little chart, and this is the little chart discussing what showing what the range of S. aureus is. As you can see, it starts with the humans and it can veer out to cows, pigs, chickens, rabbits, and you get you get the gist. I would assume by just looking at this chart. Let's move forward. Next, we're going to discuss the morphology of this microbe. And before we get into that, we're going to discuss what morphology is. Morphology is the study of the features that distinguish closely related species of organisms, and this can include microbes and then as well as plants and animals. It uses the classification. This form of classification uses different features such as the color of the organism, the size, and the proportion um, of the organism to and their proportions to determine their morphological classifications. Um, like taxonomic classifications. Um, S. aureus can be difficult to classify just off of morphology alone. And so when microbiologists look at this, they have to be very specific in what they're looking at and what they're classifying. Um, Now we're going to dive in the morphology of S. aureus, and it is a gram-positive bacteria, and this microbe tends to range in diameter from 0.5 micrometers to 1.5 micrometers, and it is also just a fun fact here. It is non-spore forming, and that is compared to other different types of bacteria um that have spores. This is a one of the rare occasions where no spores are formed. And when you look at it under a microscope, as you can see in the picture here, it is in clusters, and these clusters are compared to clusters of grapes that are on a grapevine. And that's actually where the name Staphylococcus comes from. Um, and it actually comes from the Greek word staphyle, which means bundle of grapes, to be more ironic, I guess. And now from here, let's move on. Now we're going to discuss one of the biggest sections, and this is the habitat requirements for S. aureus. Um, the natural habitats of S. aureus are the hosts that they reside on. So this can be, like we discussed earlier, humans, cows, different types of farm animals, and um, where they reside inside of each species. Um, the One of the biggest requirements of SROs habitats is that they need a specific temperature range to grow, to thrive, and to reproduce. And this temperature range is from 4 degrees Celsius to 46 degrees Celsius. And there's also a thing called an optimum temperature. And the optimum temperature is the temperature at which the microbe can grow and get do its functions most efficiently, and that optimum temperature is 37 degrees Celsius. Uh, the environment that they reside in must also have a specific pH range, and this pH range is between 4.8 and 8.0 pH. And the optimum pH range of S. aureus is 6 to 7, which, as we discussed earlier, the optimum pH range is also where it can do its functions best at. And this microbe is very unique in itself, is that it can live in aerobic and anaerobic conditions. So aerobic meaning that oxygen is present, and anaerobic meaning that oxygen is not present. Um, it grows best in the anaerobic conditions, though. So this means it grows best in the presence of oxygen. So that's when it can multiply, reproduce, divide, and so forth. Right here in the picture here is just little representation of how it resides in the nasal cavity in the nose and so forth. If you wanted to pause and take a look at it now and just study over it, as we do not have time for it today. Next, we're going to discuss the life history. Um, and Staphylococcus aureus goes through 5 different stages of development when creating its biofilms and so forth. And those different stages are attachment, multiplication, exodus, maturation, and dispersal. The first of these stages is the attachment stage. And in this stage, the S. aureus cells attach to a surface, utilizing that surface's variety of cell anchored proteins. Um, the cell wall anchored proteins are specific for each of the host matrix's substrates as well. The second stage is multiplication, and this happens after the S. aureus attaches to the host cell surface. And if they're in the presence of sufficient nutrient source, then they will begin to multiply and divide themselves, creating daughter cells. The third stage of their development cycle is the exodus stage. Now, in this stage, it's really classified as an early dispersal type stage, and it also coincides with the formation of microcolonies as well. Um, and in this stage is also mediated by a nuclease dependent degradation of the eDNA that is inside of the S. aureus cells. Um, in the maturation stage, this is the stage where microcolonies begin to form, and they provide an increased surface area, and this helps with race removal, nutrient exchange, and the promotion of dissemination of their cells to distal sites as well. And the last stage is the dispersal stage. And in this stage, the S. aureus cells are then dispersed throughout the host and throughout the microbiota of the host. And in this stage, the development process then begins over again. Thus, more S. aureus cells are then created and just spread throughout the entire body, resulting in more than likely an infection. Here to the right is just a little graph of the what we just discussed, from the attachment to the monolayer and microcolony formation all the way to the biofilm formation, and then it just restarts. You can pause now and study this further if you want to, um, as we do not have time due to our time limit for today. And lastly, we are going to discuss infectious diseases that are caused by S. aureus, and one of the most common is cellulitis. And this is a type of skin infection. And in this infection, inflammation is spread throughout the skin and as well as the tissues that lie directly underneath the skin. And this can be very, very painful, very uncomforting, and so forth. And this one of the main causes of this infection is, let's say you get like a small wound on your leg or your hand, and you decide not to treat that wound with antibiotic medication, and this can be things such as like Neosporin, or if you do not clean the wound out, this can result in the infection, causing the spread of cellulitis throughout. And people who have this infection, um, they are they often experience symptoms such as nausea, chills, and as well as the infected area becomes heavily inflamed, So that would cause it to become tender to the touch, thus resulting in a very painful experience. And this is one way they treat this is antibiotics, or if it gets worse to where there's swelling on the leg, or let's say you get it on your leg and there's swelling in the infected area, they will cut it and let it drain out and treat it with antibiotics and keep it wrapped um, until it is fully healed. And um, this concludes our lecture for today. And like I said, this is our final lecture. So I hope you all have a great rest of your semester and a great rest of your year. Thank you for joining, and goodbye.